Okay, welcome to this week's lecture, which is on methods in microbial ecology and the microbiology of the built environment. Because, we spend so much time in microbiology really focusing on the medical aspects of microbiology and various pathogens and whatnot. But there's a lot more beyond that, especially with regard to the environment. And ultimately understanding this ecological microbiology further kind of enhances our understanding of the human and medical microbiology as well. So first you have to ask yourself, what exactly is microbial ecology? And, so many times when students think of ecology, they basically just think of it as, oh, that's trees and the outdoors, so we must be looking at bacteria on trees. But microbial ecology is so much more than that. It's the interactions between various microbes, so between different bacteria or different fungi, as well as their interactions with the environment around them, including plants and animals and even us. And these interactions, or the concept of microbial ecology itself, they all include things like symbiosis, biogeochemical cycles, and interactions that can affect things like pollution and climate change, which are big topics nowadays. Okay? And so when you look at this figure here of microbial communities and the various interactions or concepts that we can study surrounding them, the first one we're going to look at is community structure. Okay? As it says in this figure, kind of trying to figure out which microbes are present, in what abundance, and various composition and structural similarities. Now, in order to study that community structure, there's various things you can use, such as enrichment or isolation culture dependent. Okay, and so we're going to go through that now in the following slides. So as I mentioned, what we're looking at here, these culture dependent analyses, these are different ways to study the community structure of microbes, okay? So that's within microbial ecology. And the two main things that you see here are enrichment and isolation. Now, I want you to know the difference between these two terms. When we talk about enrichment culturing, what that means is you are going to select for the desired organisms through manipulation of media or conditions, okay? So basically think of dissecting a microbial community from nature, and you end up with this robust collection of organisms because you're using growth conditions that favor certain variety of microbes, okay? So basically this is a technique that can help you enrich what might usually be scarcer or lower concentration microbes. Okay, so when you hear enrichment, think of culturing or using conditions to get robust, a whole lot of growth of different microbes. And the big example is the Winogradsky column that we're going to see on the next slide. The other way to study community structure is through isolation. So now, instead of looking for robust variety of microbial growth, with isolation, it's as the term suggests. You want to isolate or select pure cultures of single organisms, and you've all done this in the laboratory, okay? An example of isolation technique to study community structure is the quadrant streak that you performed. Okay, so rather than trying to get a robust variety of different microbes, with the quadrant streak, you take a sample and you dilute it down to get pure single colonies, individual single organisms. Okay, So make sure you understand the difference between enrichment and isolation. And just in case you missed what I was saying, I'm going to have a recap slide now so that you can pause and take notes. Okay, so here are the recap notes so that you can jot down what I was saying before. You can pause the video and take down the notes. Now, here is the Winogrodsky column that I mentioned as an example of enrichment. So again, remember, when you hear enrichment, think of the example Winogrodsky and visualize it as a way to get a whole lot of different growth, robust growth. And, that's what you should ask yourself. Oh, so what's the purpose of a Winogrodsky? To get robust enrichment of a variety of different microbes. And basically what they do is they take this, you can even use a bottle, basically. So they take this clear vial or an empty soda bottle sometimes, and they pack into it various things, such as some organic material. Sometimes they shred up some paper and whatnot. They might put some soil in it and various different, water samples and whatnot. And in this Winogrodsky column, so here you see what they put into it, you end up with this gradient. Okay, because if you think about it, the top of that column is what has exposure to air and the most light and whatnot. So that's where you'll get microbes that have the need for a lot of oxygen, a lot of sunlight. And as you go deeper into this column or bottle toward the bottom, well, now you're going to get to see a different robust variety of organisms, the ones that thrive in low oxygen, in low sunlight. Okay, so things like fermenters or anaerobic microbes. Okay, so it gives you a wide gradient of different organisms, nicely enriched. Now, the thing with doing enrichment is that you end up with an enrichment bias, meaning that the most rapidly growing organisms in that condition are going to dominate. And that might be different from the real ecosystem where that same organism may not really be relevant, okay? Because the nutrients available in the lab culture are typically going to be much higher than in nature, where, there's kind of that limitation of nutrients and they're not just given food, they have to kind of search it out and find it. So the enrichment bias ends up having what you may call weed species, kind of like weeds in a garden. These weed species of microbes, they'll grow really fast, but they're actually insignificant to the normal ecosystem environment, okay? So sometimes you might want to have to do certain dilutions to kind of reduce their abilities. But when it comes to setting up enrichment studies or cultures in a lab setting, even though they are valuable to kind of visualize different robust organisms, you have to keep in mind that ultimately a natural habitat is always going to be nearly impossible to create in a lab. So make sure you kind of think about, if you're setting up, for instance, a Winogrodsky column, which is an example of enrichment, well, how is that going to have bias within it? What are some things that you can't replicate in a Winogrodsky? Things that the microbes would encounter in the environment that they won't encounter in that bottle that you set up? Well, think about all the plants, all the animals, even us that are missing from inside that bottle. Okay, so enrichment is a valuable technique in some senses, but you also have to remember about the bias and kind of remember the reason why enrichment cultures are not going to be a perfect representation of the ecosystem. Okay, so make sure to put little stars next to that question of the reason why there's an enrichment bias, and why is enrichment not going to be a perfect representation of an ecosystem. Okay, so here we have a slide to look at isolation techniques. And within isolation culturing, you can either have streak plates or dilutions. So this first figure right here, this should look very familiar to you. So when I ask what technique is demonstrated and what's its purpose, well, that is the quadrant streak that all of you performed. And the purpose of that, if you remember, was to take a concentrated mixed culture and dilute it down in each quadrant so that you end up with these individual pure colonies representing an individual organism. Now, the bottom one here you haven't seen before. These are also a series of dilutions, kind of like you would do in the quadrant streak. But in these, the reason why they look kind of like they're suspended in air almost, these are dilutions of cultures that have been solidified in agar under what we call a paraffin mineral oil seal. Okay, so if you're diluting cultures and trapping them in with an oil seal, ask yourself, why would you want to do that? Which kind of organisms would you need to trap into the agar with an oil seal above it, meaning blocking any oxygen? Okay, so as soon as you hear that, what does that tell you? This bottom one here, this is to culture anaerobes. Okay, so this is also to dilute samples down and obtain individual cultures, but in this case it is for anaerobes. That's how it's differing from the one above. Okay, so make sure that you remember that two examples of isolation are quadrant streaking and dilutions, which can be performed on anaerobes. Now here on this slide, what we have is called the most probable number technique. So basically what we're saying here is, besides having methods of isolation for pure cultures, serial dilutions like the one that you saw in the previous slide for those anaerobes, these serial dilutions can also be valuable in estimating the number of microbes in a sample. Okay, so what you see here is you have your main enriched culture, which is any culture where you just grew some bacteria in liquid media, and now that the culture is cloudy, so you know that there's growth in there, but you don't know approximately how much. Okay, you can't just look at a cloudy culture and say, oh yeah, there's 1 million bacteria in there, or 2 million bacteria. But what you can do to estimate how concentrated that culture is, or how much bacteria is actually in this container, what you do is you take some of it out, so in this case 1 ml, and you dilute it down into media, or sometimes we use water. So you take 1 ml and add it to 9 ml. So for that, to calculate the dilution factor of what you just did, you do 1 ml that you put into the dilution, and you divide by the now total amount in that tube. So you would say 1 ml over 10, because you did 1 ml plus 9 ml. You would then take 1 ml out of that first tube and put it into a second tube. So now you did 1 ml into another 9 ml, but what you have to do here is you have to keep in mind that that 1 ml that you put in, that already had a previous dilution. So now you have to do your 1 ml over the total of 10 mLs, but multiply that by the previous dilution, which would be that 10 to the minus 1 over here. You then dilute the sample again. So you take 1 ml out of there and put it into here. And now that's 1 ml over a now total of 10 mLs, times that previous dilution, which is 10 to the minus 2. Okay, so I'm going to write this out on the next slide as recap notes for you to have the official calculation of how to do this. But notice each time that I'm trying to calculate a dilution factor, what I'm doing is I'm taking the amount added into the new tube, so amount added over total amount now in that tube, and I'm multiplying by any previous dilution. Okay? Then at the end of all this, you have to be able to estimate a bacterial population. When you do that, the final unit you're going to have for a bacterial population is CFU per ml. What that stands for is colony forming units per ml, meaning the number of colonies that you obtained over ml. Okay? Now, the way that you do this is, for each of your dilutions, your serial dilutions, you would see how many colonies you're able to get if you plate different amounts. So usually what we would do is, let's say we take this tube here, and I'll take 1 ml out of it and plate it on an agar plate. And then I'll take 0.1 ml out of it and plate it on an agar plate. And I'll see, with plating all of these dilutions at different volumes, 1 ml or 0.1 ml, my goal will be to try and get a plate that has between 30 and 300 colonies. And then once I have a plate with 30 and 300 colonies, I make note of how much I plated on there. Was it 1 ml or 0.1 ml? And which dilution did I take that from? Was it the 10 to the minus 1 tube, the 10 to the minus 2, 10 to the minus 3, and so forth? Okay, now the reason you want between 30 and 300 is if you go below 30, think about that. You have to do calculations with these results. If you get a sample size that's below 30, that's statistically inaccurate. Okay, that's not very good for biostatistics. If you have a plate that's above 300 colonies, well, why is that a problem? Well, more colonies to count increases the chance of human error. Because think about it, if you have 2,000 colonies to count, that's going to be a problem for you. It's not guaranteed to have very good results. Okay, so I'm going to map this out on the next slide, and then I'll give you an example as well, just so that you have notes and you know how to do a practice problem with it. Okay? Okay, so here you have the recap notes to show you the calculations. And on the bottom here, I put 2 examples of how you might encounter this kind of question. So for the first bullet point, I could ask you, okay, if you plated 0.1 ml of a 10 to the minus 4 dilution and observe 140 colonies, then how would you estimate the population? And so what you would do is you take your number of colonies, which is 140, and you write that on top of, so 140 over 0.1 times 10 to the minus 4, because these dilutions led to that amount of colonies. And then you would have to make sure to write CFU per ml next to it, because you need to have your final units. Now keep in mind, I would not then have you solve for that. I would just want to see that you know how to write 140 over 0.1 times 10 to the minus 4, making sure both of those are on the bottom of the fraction, and then writing CFU per ml. Now I could be even trickier and ask it the way that I have the second bullet point, which means that you would have to calculate the dilution as well, rather than be given a dilution. So in this one, what you would do is you would still take your colonies on the top, and on the bottom you would have 0.1, and then you would have to multiply that 0.1 by how you would calculate dilution, which you know from the previous slide is 1 ml over the now total of 100 ml times the previous dilution of 10 to the minus 2. Okay, so it would be 115 colonies over 0.1 times the dilution, which you would represent as 1 over 100 total, times the previous dilution of 10 to the minus 2. Now, if you're having any trouble with these calculations, then simply send me a remind message and I'll give you some more practice problems for you to write out. Or after lab, I can show you exactly how you would draw out any kind of problem like this. Okay. Okay, so now here on this slide, we have another form or another example of an isolation technique, but of course this one is much more complicated and requires much more expensive equipment and training than the quadrant streak, for instance. So this isn't one that you would be doing in a classroom lab. So selective single cell isolation, called flow cytometry, is an example of, another isolation technique. And in this case, it's a technique used to analyze the physical and chemical characteristics of particles that you have in a fluid as you pass through at least one laser. So you see in the image here, you have the laser passing through the fluid sample. And ultimately, what happens in this case is these cell components get fluorescently labeled, and then they're excited by that laser that we just mentioned to have them emit light at varying wavelengths that you can detect. Okay, so now you don't need to know the details of exactly how flow cytometry works because this is not like a biochemistry course. But what I want you to know is that flow cytometry is a technique that does exist to help with single cell isolation. It's able to give criteria such as size and shape, as well as some properties of the excited particles. Okay, but like I said, you don't need to know the details of exactly how this works. Another important part of microbial ecology is the ability to stain. Okay, so we've mentioned staining methods in lab before, but these are a little more detailed kind of stains that are performed here. There are general staining techniques such as DAPI, acridine orange, cyber green. Okay, and what these guys do is they fluoresce under UV light. And they're really valuable for what we call the enumeration of microbes in a sample. Okay, so basically visualizing and getting an idea of how robust or abundant certain microbes are. And these guys are non-specific ones. Okay, so DAPI, acridine orange, cyber green, the ones I have on this slide, are examples of non-specific stains. They'll stain nucleic acids, but they can't differentiate or distinguish between live and dead cells. So you won't know if what you're looking at are living microbes or dead microbes. What's important about these kind of analyses is these are now culture-independent microscopic analyses of the microbial communities that we're talking about. Okay, so you're not having, you're not studying these microbes in agar and broth. You're using microscopy now. Okay, so it's culture independent. You're not using agar or broth. And in these, like I said before, keep in mind that these are examples, a lot of times, are DNA staining, which is non-specific because all the microbes in the sample, they have DNA, they'll get stained. But like I said before, you can't distinguish between living and dead or get as much detail from these guys. Okay, but it is valuable. Any of these general staining techniques are valuable to estimate, the cell numbers present in the sample. Now, when you want to get more specific, then the staining techniques that you can use are ones like FISH. And FISH stands for fluorescence in situ hybridization. So now what they do here is they frequently target rRNA sequences of the ribosomes, and they'll stain with probes without lysing the cells. So they're not killing the cells in this process. And there are so many ribosomes that you can get a very strong signal with this. And the target sites in rRNA that are variable between different organisms allows this technique to then be very specific. Okay, so by targeting rRNA, it's valuable because you can get strong signals, but you can also get specificity to distinguish between different organisms. So for instance, you can have phase contrast, where you can use prokaryotic probes versus eukaryotic probes. So you can see bacteria versus yeast, because remember, bacteria are prokaryotic, whereas eukaryotic cells include yeast. So if you look at the pictures on this slide, in this sample, the yellow stain is a universal stain. So notice that the bacillus, the rod-shaped bacteria, and the yeast cells, these little circles, these budding circles, they're both visible in the yellow stain. Whereas when you use the red probe, which is a probe specific to eukaryotes, notice you're able to distinguish just the yeast, none of the prokaryotic bacillus. Okay, so you can use multiple probes at once, or you can use individual probes to get a valuable differentiation between cells. Okay, so please remember that general staining such as DAPI, acridine orange, cyber green, that is non-specific, whereas staining such as fish is now specific and can distinguish between cells. Now, this specific fish technique is very valuable in microbial ecology because you can kind of do things like analyze wastewater treatments. Okay, so in figure A, okay, this guy here, the nitrifying bacteria, meaning the ammonia oxidizing ones, are red, whereas the nitrite oxidizing ones are green. So you can distinguish between bacteria that have different metabolic processes. And then in this one here, you have different probes identifying a particular group of what we call proteobacteria. So for instance, distinguishing among E. coli, Salmonella, Vibrio, all different things like that. So you can see on this slide, you don't have to know the exact probes used, but that fish can really help you distinguish among different bacteria, and you could have different color probes all on the same slide. So notice in that second one, we have a robust variety of probes, and you can really see different bacteria there. So now continuing the idea of culture-independent analyses. Remember, culture-independent, we're not using agar or broth now. The first culture-independent technique we mentioned was microscopy and staining. The second one is genetic analyses. So as you see here, we're still trying to analyze a microbial community. For instance, think about, microbes in a pond sample, or that wastewater we mentioned a minute ago. So of course, the first big bullet point here is PCR when we're talking about genetic analyses. And we've already gone through that previously, but PCR, think about that as the ability to amplify different microbes, different pathogen genes. You can detect various different species, or you can detect different enzymes and products that you're looking for with PCR. Then you have DGGE, and DGGE stands for denaturing gradient, gel electrophoresis, and that's to further resolve what we call phylotypes, and phylotypes are just variants of genes. And what this technique does is it separates genes of the same size that differ in denaturing or melting profiles, okay, because their sequences are different. Okay, so basically it's denaturing the genes and trying to distinguish the different microbes. There's also TRFLP, okay, that's terminal restriction fragment length polymorphisms. I know that's a ton of words in one mouthful, but basically what this does is there'll be a terminal dye label that you put on genes, and then you cut with restriction enzymes. And this will show our RNA sequence variations in a microbial community sample. Okay, and you'll see differences in the same genes of different organisms. But ultimately, both of these techniques that I just mentioned, they both measure single gene diversity. So you can kind of get information on evolutionary history and relationships or things like that. So it's particularly useful in any phylogenetic analyses, which is like when they make those genetic trees in evolution and trying to figure out which microbes are related to which other ones, for instance. Okay, so I'm going to give some review or recap notes on the next slide. Okay, so here are the recap notes. You can pause the video and take down the notes because, like I said, I know that's a lot of information. Okay, so the previous slide was showing single gene analysis, basically looking at various microbes in a community, such as a pond, and looking at the differences and similarities of a single gene, let's say gene A, okay? Now this slide is to show you that also what exists is environmental genomics that are metagenomics for total community genetic analyses, meaning you can have single gene analysis like the previous slide, or you can have total gene pool analysis. So now you're looking at all the genes in that pond, for instance, not just one, gene A differing from, microbe A, B, C, D. Okay, so in this case of metagenomics, looking at the big picture, now what you're going to look at is DNA clones from the microbial community that get sequenced, and then you detect as many genes as possible to give the gene pool picture. And you can even detect genes that you wouldn't have amplified by current PCR primers. Okay, you're grabbing all genes, not just specific ones that you're targeting. And this is especially useful for assessing what we call phylogenetic or metabolic diversity of an environment. Basically trying to see, okay, in this pond area, what are some of the various enzymes that these microbes are capable of producing, versus another pond or a lake, for instance. How do their genes and various microbes vary from location to location? Okay, so keep in mind that, as you see in this figure here, you could have the small community sampling approach with just single gene analyses, or you could have the big metagenomics, environmental genomics approach of just grabbing every gene in that location. So we call it total gene pool analysis. Now, what's interesting is, in addition to metagenomics, which is looking at all of the DNA in that community, there's also metatranscriptomics and metaproteomics. And you could ask yourself, well, what do each of these mean? Well, metatranscriptomics, when you hear transcription, what do you think of? Well, that's making RNA, right? So metatranscriptomics is going to be like metagenomics, but now instead of looking at DNA in that community, you're going to look at all of the RNA in that community, which means all of the expressed genes, okay? Now keep in mind, that means there's a lot of genes that won't be represented. You're looking at only the ones that are getting expressed, okay? Which ones are active in that community. And ask yourself, well, if we're looking at RNA, how do you analyze RNA? Well, that's reverse transcription, right? So you have to use reverse transcriptase method. Now, the key difference is metagenomics will give the functional capacities of a community, meaning what genes does that community have? What do they have the potential to do? Whereas metatranscriptomics, now that's saying exactly which genes have been expressed, not just the potential, what actually got expressed, and what are the relative levels of that expression at a particular time and place. Now, beyond that, the next step would then be looking at metaproteomics, which is, of course, going the protein level. So now you're looking at the diversity and abundance of proteins in the community, which, like I said before, is very valuable when you consider that what are some proteins? Enzymes are an example of proteins. So that could give you valuable information about that community. So this is kind of important because mRNA have different half-lives and efficiencies of translation. So metaproteomics gives you the ability to have a direct measurement of cell function, okay, of the cell function of the organisms in that microbial community. Okay, so when you hear metagenomics, you're looking at the DNA of all the microbes in a community. Metatranscriptomics, you're now looking at the RNA, which tells you the actual cell function, not the potential that metagenomics gives. And then you have metaproteomics, which is the protein level. Okay, so again, direct measure of cell function. So again, this was a lot of information at once, so I'm going to put a recap slide next. Okay, so here are the recap notes so that you can jot down the differences between metagenomics, metatranscriptomics, and metaproteomics. So again, you could just pause the slide here and jot down the notes. Now this slide here is just a visual summary of some of the analyses that we mentioned. So in this first area, you have looking at nucleic acids. This can also include some of the stains that we mentioned, such as DAPI and acridine and cybergreen. You also have a look at the RNA versus the proteins. So again, remember that when you look at anything dealing with DNA, you're looking at the functional potential of that microbial community, all the genes they have, so there's a lot of potential there. Whereas looking at RNA and proteins lets you know exactly what has been made. When you see metabolites, that's just looking at metabolic products, kind of like you'll see in lab when you look at things like fermentation products or breaking things down. Now here what we have are some chemical assays for measuring the microbial activities in nature. So in one case you can check for lactate or lactic acid and a reduced product of H2S. So it's like when you're looking for oxygen reduction, for instance. And so you can see if lactate oxidation is occurring by the microbes in sediment, for instance. And you do that by adding lactate to the sediment and then analyze the consumption. So you see that in this first image here. So one of the ways they measure microbial activity, you put that lactate in and you look at how much time before that lactate has completely been used up by that community. Okay, and as that's getting used up, you have that reduction product of H2S building up. Okay, so that kind of gives you an idea where those curves are. Did the lactate get used up very early on? Gives you an idea of how much activity you actually have in that microbial community. Alternatives can also be using radioisotopes. Using radioisotopes when there's very high sensitivity required or when you need to analyze turnover rates. So, the turnover within that microbial community. Again, we're not going to go into all the details of the exact reduction and processes occurring here. Just be aware that if you wanted to measure microbial activities in nature, that you would either use chemical assays, such as looking at lactate reduction, lactate oxidation, or using radioisotopes. Now, the last part of this lecture that I want to go through is microbiology of the built environment. And so the built environment, you have to first ask yourself, well, what does that mean? Well, when we talk about built environment or built systems, that's what mankind has created. You know, we took this environment that was once trees and just lakes and ponds, and now we've created all of these infrastructures, like for drinking water and waste management and all these factories and whatnot. And by creating these kinds of things, in one sense, we've kind of destroyed some microbial habitats, or even when you think of animal and plant habitats, to put these things up. But at the same time, you've also kind of created new microbial habitats. Okay, so when you think of all of these built systems, these infrastructures, you kind of have to think of what's going on. So an example of not favorable is the corrosion of pipelines. So natural process of microbes, is simply exploiting the resources provided by the built environment. So you've now put all of these pipes all over the place, and microbes love to oxidize metals. And when they oxidize metals, they produce acids that further corrode those metals. So basically, by building these new systems, by building these pipes everywhere, you've created some, microbial habitats that's favorable to the microbes. They love those pipes to break down, but not favorable to the humans, because you didn't realize that what you were building was perfect for microbes to then exploit what you put up. Okay. Now, with that built environment, you can actually basically exploit microbes, because microbes, we can look at them as Earth's greatest chemists. So hopefully we're not offending a certain chemist in the class, but yeah, microbes are far better than any human chemist. And one example of how we can use them and their chemistry abilities is that we can have them help extract things for us. We call it microbial leaching. So you can have the removal of valuable metals, such as copper, from things like sulfide ores by microbial activity. So microbial leaching is pulling out or breaking down these sulfide ores to gain valuable metals for us. And so basically what's happening is you have the promotion of acid production and the breakdown of sulfites by what we call acidophile bacteria. They like that acidic kind of conditions. And you can use this in large-scale mining operations. And they're basically like little microbial miners for you. They're breaking down these big ores, and they're basically extracting what you want from them. And so you'll see these giant tanks of, containing mixtures of acidophile bacteria that then solubilize the minerals and release metals as a product. So this is used for releasing metals such as gold, copper, iron. And no matter which ones of these metals you're obtaining, we call it microbial leaching. Okay, so basically using acidophilic bacteria to break down minerals and ores that are low grade and produce very good metals for us. Okay, so another example of how microbes can be used by us in our built environment is what we call bioremediation of organic pollutants. So basically, we could use microbes to help clean up contaminants in our environment. And when we talk about this, it's basically because microbes, as you know, they're great at metabolizing things. They have metabolisms that are unique to them compared to us. And one of the terms that I want you to know is xenobiotics. Okay, right here I'm highlighting it, xenobiotics. What xenobiotic compounds are synthetic chemicals that are not naturally occurring in the environment, basically meaning man-made chemicals. So examples of those would be things like pesticides, biphenyls, munitions, dyes, chlorinated solvents, any of those. Just remember that xenobiotic compounds are synthetic chemicals not naturally occurring. And the big problem with them is many of them degrade slowly. So they're just chilling in the environment, causing problems for our various animals and plants. So we can use microbes to help break them down, either anaerobically or aerobically. And you end up having these microbial cleanups of environmental pollutants because the microbes, they can either degrade these xenobiotics or at least change the mobility of them to kind of contain them. So picture the bacteria kind of trapping some of these xenobiotics. Okay. Now, this is, like I said, this can be either anaerobic or aerobic. So for instance, the example on the slide is dechlorination. Sometimes they're using oxygen to do this, sometimes they're not. And it's ultimately important to kind of know that these things exist and how some of them are anaerobic, some are aerobic. It's important because of how rapidly polluted habitats tend to become anoxic, meaning lacking oxygen. So, this idea of having the ability to have anaerobes do this as well and break down and contain these pollutants is very powerful ability for cleaning up. Now, the last things that I want to mention over here is the idea of wastewater and drinking water treatment. Okay, so first of all, we have the idea of why is it so important to ensure water purity? Well, think about it. You need water. Everyone needs to drink water. And so if it's contaminated, then that could be very dangerous. And the word that I want you to remember is potable. You want water to be potable, which means safe for drinking. Okay. Now, a lot of times, you may think, oh yeah, my water's safe to drink because we have all this wastewater treatments and everything and drinking water treatments, but there are still a lot of countries, especially third world countries, where these treatments are not that great. They're not that advanced and they have problems. And when you think about treating water and purifying water, you have to ask yourself, what are the most common bacterial contaminants? Well, the most common bacterial contaminants, as you get to see in lab, are what we call coliforms or enteric bacteria. And where would you find coliforms or enteric bacteria like E. coli? In the GI tract, not just of humans, but of any animals. So when you think of water contamination, you have to remember that a lot of it, as gross as it sounds, is because of poop from animals. Okay, so you're not going to picture a guy or a woman using the water as a restroom, but think about all the fish in there. Think about all the birds that fly over these waterways and these treatment facilities and all. So it's very easy for animal fecal contamination to occur. So when you do any kind of water testing, the big thing you're going to want to look for are coliforms, specifically E. coli, because if those are present, then there has been some sort of fecal contamination, and there's probably other bacteria then present as well. And the way you can test for these things are things like lactose fermentation, so looking for red media changing to yellow, and gas production. using EMB plates. And EMB plates will get a beautiful shiny green metallic colored colony growth if E. coli is present. You could do gram staining because we know that E. coli or coliforms are gram-negative bacilli, which means if you gram stain them, what will they look like? Well, they'll look like pink rod-shaped bacteria. Okay, or what we use nowadays is coliscan plates. And if you see blue colonies, blue tells you it's E. coli, and pink will tell you it's other general coliforms. Okay, so there's various ways to test. And ultimately, what you want to make sure to then do is once you know that there are, purity issues, then you need to treat and purify. And the funny part about treating and purifying is that you can even use microbes to actually treat the water as well. So you basically use good, helpful microbes for treating the water to clean it up. And so, as we saw in previous slides, the idea is microbes are great at degrading pollutants. They're very good at digesting things that, we wouldn't be able to. So for instance, as you see in this figure here, wastewater, you can have screening and sedimentation. And then over here, these are microbes that are basically breaking down the sediment and digesting it into cleaner products, into, more broken down products. Now this slide here is to show how some of the steps for purification of wastewater. So like we mentioned, you have the microbes that can help you out. You also have the idea of sedimentation, coagulation and flocculation, filtration, and disinfection. Okay, so when you see any of these terms, you should kind of generally know what they mean and that they're for cleaning up wastewater that you want to be or to end up being potable, which is safe enough to drink. Okay, that's what potable means, safe drinking water. And so these further purifications include sedimentation to pull out, certain larger particles, and so you can see that first step of sedimentation there. And then when you hear coagulation and flocculation, what that means, if you flocculate something, I know I love that word, it sounds so weird. So flocculation is basically aggregating or clumping up things to trap the microbes and get this suspended matter, okay, so that you can then better remove or filter, because if you flocculate the particles, you get these clumps that will then settle out through filtration, so just the water going through. And then lastly, you can use chlorine or chlorination to help disinfect, okay, because chlorine gas is very, very good at sterilizing. In fact, a lot of times that can even kill endospores. So it's a nice, powerful technique, okay? So basically, this just shows that there are multiple steps to get rid of any kind of contamination and to really help inhibit any further growth that may occur. So unfortunately, that's not the end of the story there. This idea of cleaning up the wastewater treatment, it's not a perfect science yet. And a lot of drinking water ends up being associated with diseases that usually don't even get reported, because a lot of people don't realize that it's their drinking water that sometimes can make them not feel so great. And a big part of this problem, the reason why drinking water can still cause you problems, is the amount of distance that water then has to travel once it's been purified to get to your house. Look at, think of all the pipes that it's got to make its way traveling through this far distance from where it's been cleaned up. Now, sometimes these problems can be as mild as, just minor taste or odor problems, but sometimes it could actually be that, growth of pathogens is promoted during this large distance of pipes away from the water purification plant. So as this water is trying to make its way to your house, ultimately these long distances of pipes, they select for pathogenic bacteria. And think about it, what does opportunistic pathogens means? It means that, bacteria that normally might not be a problem, might be in very low concentrations, well, now they have a beautiful resource-rich environment to grow. Competition has been taken out, and they can be problematic, and they can get you sick. The other big problem with all of these pipelines for bringing that water to you is they ultimately select for resistant bacteria and biofilm growth. So what that means is you get biofilm and resistant growth because the further this water has to travel away from the water purification plant, that means that the further distance, there's a reduction in the chlorine concentration. So reduction in chlorine concentration with increased distance from that water purification plant means that there's going to be more tendency of microbes then popping up and forming biofilms all along the pipe walls. And if you remember, biofilms are when different bacterial species, they clump together and they stick to a surface. And they're very tough to get rid of because they're all different species working together. Now, if you think about it, you're like, well, fine, why don't we do something to get rid of these nasty biofilms people could get sick from? Well, complete elimination, that would require complete nutrient removal and maintaining the proper amount of residual chlorine levels through all of these pipes, these crazy distances of pipes that are all over the place leading to houses. Because think of it, how far away is that water purification plant? I'm fairly certain none of you can look out your window and see a water purification plant. That's how far away it is. That's how long these pipe distances are. So the idea of complete nutrient removal and maintaining proper chlorine levels, doing either of those is not really realistically possible. And so ultimately growth is always going to be unavoidable. But what I want you to really understand is that even though this growth is possible, they do, try to minimize it. The water purification process is a very good process. And a lot of times, depending on where you are, you do always have access to, purchasing water or buying filtration systems for in your house and everything. Okay, so now as always, it's your turn to be the professor and head over to the discussion board, which I wrote specific instructions for on how many questions to write. Okay, and as always, make sure that if you have any questions, you need a little extra help with any of the slides, or if you want extra practice problems, especially for the dilution and the estimating bacterial populations, just send me a remind message and I will be more than happy to help you. Okay, thank you.